Assalamualaikum warahmatullahi wabarakatuh, everyone. So this is pre-recorded lecture for week 9, titled Techniques in Biochemical Analysis. This is chapter 5. So in this chapter, we will cover the extraction methods 5.1, 5.2, chromatography principles and techniques, 5.3, spectrophotometry. But for this week, week 9, I will cover only from 5.1, 5.2.1 until 5.2.2. Okay, so next week 10 later on cover on the others lah, the other chapters. Okay, so extraction methods. Okay, so in biology, okay, we also need to analyze, okay, the chemicals inside organisms. Okay, so organisms, we have two types, animals and plant. Okay, so to understand the compounds, okay, that presence inside animals or plants, okay, we need to extract the samples. So the importance of understand the presence of the compounds, okay, from the organisms yang kita nak study is that we know, okay, with the knowledge of understanding the compounds present from the organisms, it will help us to understand the reasons of the biochemical processes yang berlaku, okay, or the biological activity that resulted from our research. So that's the importance of the chemical analysis, okay, so that kita boleh proven or explain, okay, the data finding from our biological activity. So to do the biochemical analysis, okay, the first method, the first technique is that we have to do the extraction method. Okay, so what is extraction method? Extraction method is a technique to separate, okay, the compounds, okay, from the complex matrices, or also known as transfer, okay, the compounds from a solid, or solid to will represent the organisms such as the plant or animals tissues. Okay, so we want to transfer the compounds from the solid particles, okay, into or liquid into a different solvent ataupun phase. Okay, so solvent kena identify lah what type of solvent, whether it's a polar solvent or non-polar solvents. Okay, and the techniques of extractions and to identify the types of solvent is that we can refer them from our reading from articles or journals, okay, or for previous studies. And then the techniques of extraction is that we could use these three types of techniques. First is the distillation, okay, second is by solvent extraction, and number 3 by solid phase extraction. So in distillation is more the important key that you have to know about distillation is that they involve the process of boiling and condensing. Okay, what is boiling? Boiling tu didih, and condense tu maksudnya you pindahkan the from it's a process of changing, okay, from the vapor kan, after boiling kan dia vaporize kan, okay, the solvent will be vaporized, and then the vapor then after undergo condensation dia akan change from vapor into liquid. Okay, so the process of distillation is process of kita boiling, change the liquid into vapor, and then condense, okay, means liquidify the vapor, okay, means the vapor change into liquid. Okay, so the important objective of having distillation is that to separate, okay, and purifying your liquids. Okay, so distillation involve again, okay, the process of purifying and involve process of boiling and also condensing the vapor. Okay, the important the uses of this technique is to separate the volatile and the non-volatile substances. Okay, and to separate two or more volatile liquids by exploiting the different boiling temperature liquids. So we expect, okay, that the volatile will be vaporized first lah. Okay, so the by understand the boiling temperature of the liquids. Liquids ni referring the solvent yang kita guna untuk extract our samples. Okay, so kita gunakan this by understanding the boiling temperature for the different types of solvent, sebab different solvent they have different boiling temperature. So yang more volatile biasanya dia akan have low boiling temperature, dan less volatile dia akan have high boiling temperature. So kalau kita guna at low boiling temperature, the more volatile will vaporize first lah. Okay, so the principle of distillation is that kita guna the liquid with lower boiling point, dia akan evaporate first, okay, and then the vapor then will be cooled and condensed menggunakan condenser. So important aspect for distillation is that this is the most common separation technique. Okay, kalau you baca pun mana-mana journal, okay, this distillation is a very common technique used, okay, to separate to separate the compounds, okay, from the solid or liquid into solvents, kan, into solvents. So this is the most principle of extraction yang being applied, okay, the distillation process. Okay, then this techniques consume very much energy because dia melibatkan cooling and heating, kan. So the heating tu dia involve high heat, kan, kena boilkan dia, kan, and then kena guna water pula untuk cool down the vapor, so dia boleh condense into liquid balik, kan. So it consume lots of energy, this techniques of distillation, okay, this extraction technique. Okay, here example of the boiling of water. Okay, so it will form it will vaporize, then the vapor then will condense, okay, by helping of the cool temperature, okay, by the presence of water tadi, nanti at the condenser menyebabkan dia boleh liquidify lah, so the vapor will change into liquid. So this is a process of distillation, class. Okay, and distillation have two types, simple, another one is the fractional distillation. Okay, distillation. So this is the first type of distillation. The first one is the simple. In this simple distillation, okay, you consist of, you boil the liquid or your solvent tadi, okay, and then the solvent then dia akan condense, okay, dia akan condense, means dia akan change lah, vaporize tadi dia akan membentuk liquid, okay, that is condensed lah, okay. In this process, okay, dia akan collect the distillate ataupun yang liquid yang telah berjaya ditukar selepas melalui condenser tadi. Selepas dia sejukkan tadi kan di liquidify, dia akan bentuk liquid. So the liquid then will be collected in a separate container. Okay, dia tak bergabung balik dengan solvent tadi. Selepas solvent tu dididihkan, okay, dididihkan solvent and then dia akan vaporize. Vapor tadi dia akan undergo condensation dekat condenser, dan the liquid yang berjaya dihasilkan after condensation tadi akan di-collect as a distillate dekat separate container. Okay, dia tak gabung balik dengan solvent tadi. So in a separate container. So this in this simple distillation, mostly used to separate lah the most volatile liquid. So volatile liquid tadi dia akan more volatile, dia akan at specific boiling point dia akan vaporize, and it is more volatile, so dia akan move vaporize first lah. Okay, so so distillate yang terkumpul tu adalah the liquid or the solvent yang more volatile. Okay, so dia akan terkumpul dekat distillate sebab dia lagi cepat boil, and dia adalah lebih volatile tadi lah. Okay, so that is collected at bahagian distillate. Okay, so from this picture here you could see, okay, dia ada, this is a collection flask, okay, di mana dia akan collect the distillate yang more volatile tadi. So yang telah berjaya, yang firstly being vaporize. Okay, so more volatile components, okay, dia akan vaporize first, okay, the yellow one particles, and then dia akan vaporize. After vaporize, dia akan condensate, okay, dekat condenser. This is condenser. As you can see here at condenser, dia ada cooling water, dia akan move, the cooling water, okay, there will water from pipe, kan, dia akan give the cooling effects, okay, di mana dia akan masuk air and then keluar air dekat bahagian condenser ni sebagai menyejukkan bahagian dekat dalam condenser. So bila dia sejuk, dia bantu menukar the vapor into liquid. So the liquid dan the liquid yang the volatile compound, the component tadi yang dah being liquidify tadi akan masuk ke dalam collection flask, dan this is called as distillate. Okay, as example here, kita guna mixture of salt water. So salt and water, so water is compared to salt. Okay, water is more volatile compared to salt. So water will be vaporized first, okay, and then the water then will be collected, okay, as a distillate dekat bahagian collection flask, okay, and then salt will be the only be left lah dekat bahagian distillation flask. Okay, this is the simple distillation, okay, di mana dia hanya menggunakan condenser to cool down the vapor, and dia melibatkan apa this distillation flask, okay, by heating the samples ataupun the mixture. And then the second types of distillation, there are two types of distillation. First is the simple, second is the fractional distillation. So for fractional distillation, dia beza dia dengan simple distillation is that dia melibatkan fractionating column. Okay, kalau compare tadi, simple distillation dia hanya melibatkan boiling, big boiling flask and also the condenser, and then they collect the distillate kan. However, to this fractional distillation dia ada ketiga-tiga tu, tapi perbezaan dia ada penambahan, dia ada fractional distillation. Okay, fractional column, fractionating column. When a fractionating column, dia terdiri dengan filled with beads, okay, which help in give better separation between the liquids. So among liquids pun ada banyak pula perbezaan dia based on its boiling point. So based on the different boiling point, it help them to separate, okay, the components of the liquid. So liquid macam ni, so dia ada mixture yang besar. Dalam mixture yang besar tu dia boleh separate lagi komponen yang berbeza. Okay, nanti kita akan tengok from the picture as an example. And this fractional distillation used to separate, okay, volatile liquid with a smaller difference in boiling point. So volatile dia akan being separated, mana-mana volatile liquids dia akan move first lah compared to the less volatile compound. Okay. Okay, so this is example or the best example for fractional distillation. It involves, okay, with the burn of crude oil. Crude oil ni ada numerous of mixture of samples, components. Okay, so inside the crude oil dia pecahkan lagi the components, okay, using this fractional distillation. Okay, from the fractional distillation, okay, the more volatile component dia akan separate first lah. So volatile component ni adalah di mana dia mempunyai boiling point yang lebih rendah berbanding yang lain. So at boiling point rendah, dia lebih mudah volatile, dia akan mudah meruap dan dia akan collected first lah. So first is the refinery gas, second followed by petrol, and then lastly is the bitumen. Okay, so because of the difference boiling point, okay, and volatility, okay, this help in the separation of component from the crude oil. Okay, so this is the best example of using fractional distillation in separation of component from a crude mixture. Okay, so this is fractional distillation. So distillation only have two types. First is the simple, second is the fractional distillation. Okay, other than distillation is one of the extraction method kan, I've been mentioned just now. Okay, so after the distillation, okay, we have solvent extraction. Okay, solvent extraction is one of the technique of extraction. Okay, number two, first is distillation, second solvent extraction. In solvent extraction, a method for separating mixture by using the differences in the solubility of the components. Okay, and based on the solubility pula. Okay, dia lebih soluble ke ataupun kurang soluble. Okay, And in this tadi membahas on volatile ataupun non-volatile kan, yang lebih volatile akan come first kan, and this is based on solubility of the component, kadar ketelarutan dia, kadar ketelarutan komponen tu dalam solvent. Okay, so by using this technique solvent extraction, okay, among widely used solvent extraction yang commonly kalau kita baca adalah teknik dia by SOXHLET method. Second is by liquid extraction. Okay, in the SOXHLET extraction, okay, dia menggunakan this condenser, okay, and this is the extraction chamber. Okay, here is extraction chamber. Inside the extraction chamber dia ada letak timbal. Okay, kita panggil timbal. Okay, di timbal kita letak our sample lah yang kering. Okay, sample yang kering kita letak, for example dry leaves, okay, dry stems, okay, dry roots ke kan. Okay, kita letak inside the timbal. Timbal ni yang the white color ni lah. Okay, dia ada macam like filter paper that been shaped. Okay, that exactly boleh muat kawasan chamber ni, extraction chamber. Okay, and then dimasukkan, first dia akan masukkan the dry samples ke dalam timbal. Okay, then the timbal will be put inside the extraction chamber. Okay, in this technique, the solid extraction, okay, dia akan melibatkan proses of it involve, okay, the process of heating of the extraction solvent. Kita letak solvent kat sini without any combination with hanya solvent saja. Kita akan letak dalam this boiling flask, okay, and then dia akan boil didih. Okay, dia akan vaporize, masuk pergi ke condenser. So dekat condenser, okay, again dia akan process of condensation. Okay, from vapor dia akan turn into liquid, then the liquid dia akan passing, dia akan melepasi the liquid, dan dia akan pergi menitik-nitik, okay, pergi kepada that timbal. Okay, the timbal mana that consists with the dry samples tadi. Okay, so a water cooled condenser tadi, okay, dia akan condenses vapor, dia akan hasilkan drips of solvent yang telah berjaya dikondense pergi kepada timbal, and the solvent yang dah menitik tadi from condenser, dia akan contact, dia ada physical contact with the solid sample yang dry tadi. Okay, so bila dah terkumpulnya solvent yang menitik tadi sampai one level, okay, where the siphon tube level is reached, dia ada satu level at the siphon tube here. This is siphon arm. Okay, when it reach at one level, where it is siphon tube, okay, the solvent yang presence, okay, drips of solvent, okay, yang menitik tadi yang terkumpul at the extraction chamber, okay, yang contact with the samples at the timbal, okay, solvent tadi akan turun balik pergi ke boiling flask. Okay, dia akan return to the reservoir. The reservoir is the boiling flask tadi lah. So the process can be continued by hours until days. Okay, as long you pastikan you masukkan all the components tu directly lah, tak ada sampai dia terkeluar ke. So make sure the process is occur fluently. Okay, and it can go until days, okay, or weeks, okay, as long everything is, sebab dia solvent takkan hilang, dia akan pergi balik ke reservoir, dia akan terus vaporize, dia akan condense. Okay, the process is continuous. Okay, continuous without reducing in the number of the volume of solvent. Okay, so the components here listed from number 1 until number 11 inside the SOXHLET procedure. This is the siphon tube. Okay, this is a timbal when you add with your dry sample. Okay, and third type of extraction. Okay, tadi first this distillation. Okay, second is the SOXHLET. Third is the liquid extraction. Okay, these three are the common type of extraction applied lah dalam kita punya research. Okay, liquid extraction is that dia menggunakan based on solubility. Okay, mana yang lebih soluble, mana yang kurang soluble. Okay, so in this example, okay, dia menggunakan image here, it used a separatory hanya menggunakan the separatory funnel. Okay, so separatory funnel ni dia akan separate based on solubility of mixture lah. Okay, so here they have two substances dissolved in water. Dia ada dalam water saja. Dia gunakan dua substances dilarutkan dalam air, and then after that, okay, dimasukkan organic solvent ataupun immiscible ataupun insoluble solvent. Okay, dia letak organic solvent yang hydrophobic, yang tak suka air. Okay, and then after that, lepas dia add with the organic solvent or immiscible solvent or insoluble solvent, you have to shake your mixture inside the separatory funnel ni. You mixkan them, you mix, and then you akan nampak there will be partitioning. Okay, the partitioning dekat bahagian you punya, dekat your separatory funnel. Okay, so what will be collected here is that yang dekat bahagian hydrophobic here ni adalah yang dekat bahagian organic solvent yang you letak tadi akan terkumpulnya hydrophobic molecules. Okay, yang mana tak suka air tadi, dia akan terkumpul dekat components, dia akan terkumpul, it will collected or partitioning at the organic solvent area. And then what you have to do is that you have to remove lah mana yang you nak. Okay, you ambil lah kat sini, keluarkan dulu the water solvent, the water solvent with the component yang polar. Okay, and then barulah you take the organic solvent with the hydrophobic molecules. Okay, so that's how you separate your substances inside your samples. Okay. Alright, this is the liquid extraction. Again, this is example of organic solvent pula. Dia guna organic solvent here, and this is the aqueous solvent. So this is organic solvent with hydrophobic samples. You add water, you mix them. Okay, after you shake the separatory funnel. Okay, and then you akan nampak the separation of substances. Okay, yang mana it will the phases will separate by gravity. Okay, and then the water with the component of polar components. Okay, and the non-polar component at the organic solvent. Okay, so this is liquid extraction. The liquid extraction is one example of solvent extraction that is based on solubility. Okay, third of extraction, okay, adalah solid phase extraction. Okay, solid phase extraction di mana, okay, solvent tadi is replaced by solid particle surfaces. Okay, dia tak guna solvent, dia guna silica, powdered silica. Okay, as that is hydrophobic, that has hydrophobic functional group, and it will act as the extracting phase. Okay, dia akan jadi tempat untuk extract your samples. Okay, so for example here, this is SPE lah, solid phase. This is the apparatus applied, okay, for solid phase extraction, di mana dia letak conditioning solvent first. Okay, there are numbers of chemicals yang adalah, tapi kita tak perlu go deep into that conditioning solvent. Okay, tahulah dia ada mixture of chemicals in conditioner solvent. Okay, and then after you add the conditioner solvent, then you masukkan you punya sample mixture. Okay, then you have to add the washing solvent. Okay, washing solvent dia akan wash away, okay, komponen yang you ni lah yang you tak nak. So dia akan tinggal dengan komponen yang boleh attach dengan dia yang akan then it will wash away, okay, the component that you don't want to, and then you have to add with eluting solvent, so that this then will help in remove, okay, the components and eluting the component that you not study. And then the eluting component, then you will go to the right instrument for analytical for analysis. Okay, and this is solid phase extraction. Okay, again dia hanya menggunakan solvents yang berbeza untuk membahas washing solvent, eluting solvent and conditioning solvent during the process of extraction. Okay, so dia less of using solvent sebab because the size of this it's like a pipette, it's quite small. Saya hanya menggunakan solvent yang sedikit saja, compared macam tadi distillation, compared to the solvent extraction, dia menggunakan lots number of solvent kan, dan dia melibatkan banyak tenaga kan. Ni hanya melibatkan sedikit saja solvent, okay, sebab the size of the it's like it's a filter size. This filtration process dia melibatkan small sizes of apparatus, so they use less solvent, okay, and it is more efficient in separating your samples in your extract sample mixture. Okay, now after completely extract the samples, okay, the extraction after kita transfer the compounds from the solid or liquid samples tadi ke dalam solvent, kita dah complete our extraction. Now kita nak analyze kita punya sample tadi dalam what is the exactly the compound yang ada yang kita dah lepas kita masuk dalam solvent tersebut. So kita nak analyze dia, kita menggunakan what technique? The technique known as chromatography techniques. Okay, so as you can see in this chromatography, ada yang menggunakan filter paper lah, okay, filter paper di mana dia akan separate, okay, the compounds based on dia punya movement, okay, station of the mobile phase. Okay, kita akan tengok. So that is help the main objective of chromatography technique to separate the mixture from the solvent yang kita dah from the extract yang kita prepare tadi by extraction process. Okay, so in the chromatography technique, okay, dia nak purify our samples, kita nak separate the components of mixture inside the extract, okay, and then we will identify the components lah. So this is example of using chromatography paper. Okay, kita letak sampel kita dekat origin of pigments, letak the extract dekat bahagian line yang pertama. So this is the starting point, and then after kita dip this chromatography paper dekat dalam mobile phase, okay, dia akan allow the movement of the pigments or the mixture. Okay, it will separate them based on dia punya movement. Okay, sampailah the mobile phase reach dekat bahagian solvent line, dekat bahagian yang the end of the line. Then kita akan, and then we will look on the different of pigment color dekat dalam ada on the top on the chromatography paper. So as you can see here, okay, there are different colors, ada green, blue, green, yellow, orange, and each color refer with different components. Okay, so this again the chromatograph technique is about separating the components from the extract yang kita dah prepare. Okay, and to separate the substance in a mixture, and by the chromatography is separate them by its color. Okay, chromos is color, graphy is write, okay, to write the color and to separate the colors. Okay, and this method can be applied to all kind of mixtures. Okay, a sample that requires analysis is often a mixture of many components. Biasanya kita nak analyze our extract ni, biasanya dalam tu mesti ada banyak komponen. Memang in leaf pun ada banyak, selain daripada chlorophyll, they have the xanthophyll just now, carotenoid, kan, banyak types of components inside the extract of our samples. So it is very important for us to analyze them. So often a standard to, and then because we don't know actually what is that, kan, so kita kena refer a standard, okay, to that so that we could identify what is exactly the compound yang kita jumpa inside our extract. So often we need a standard to compare, okay, or what is a standard? Standard is a single pure compound, or it also can be a mixture of known amounts of several compounds. Okay, this used to compare with our samples. Okay, as you can see, this is a chromatogram. Okay, from our sample extract kita dapat this peak lah, 1, 2, 3, 4 and 5 peaks, and then we don't know actually what is the meaning of this peak is. Okay, actually what compound or component yang sebenarnya mewakili on each peak. So we compare with our standard. After running standard means yang kita tahu that is what Okay, for example, kita tahu number 1 is caffeine, alright, and number 2 is aspirin, and number 5 is salicylate. Okay, at that retention time kita tahu number 1 ni adalah caffeine, number 2 aspirin, and number 5 is salicylate. So bila kita run our sample, kita dapat 5 peak. Tapi based on the standard, kita boleh compare and kita boleh analyse, and then kita boleh conclude yang sample kita terdiri daripada caffeine, okay, and also aspirin together with salicylate, because the peak yang kita jumpa sama dengan peak yang at retention time, sama dengan peak yang ada pada standard. Retention time dia sama, dia keluar pada waktu yang sama dengan standard keluar. So kita boleh so that we can conclude that our samples, okay, now is contained with that kind of compounds. Okay, so that is how we want to analyze our sample using chromatographic technique. This is example of, I'm sure this is HPLC lah. Okay. Okay, so to analyze our compounds, okay, we have to identify kita punya compounds yang kita nak analyze tu based on its physical and chemical properties. Okay, for example, you memang cadang you nak cari caffeine, so you study on that physical and chemical properties of the caffeine, and then you run your samples using that using the properties of the compounds. Okay, and you have to know the solubility, the boiling point, and vapor pressure for that kind of component that you want to focus on. Okay, so this process of chromatography, other than you identify or you understand the physical and chemical properties of your compounds. Okay, yeah. And then you also have to know the state of matter of the phases yang you akan guna, the mobile phases and the stationary phases. Other than that, because with this knowledge, with this understanding of the properties, this will help you in separating the compound based on its density and size. Okay, chromatography, or known as in ancient Greeks known as color writing. Okay, they have 3 types of chromatographic methods: TLC, LC, and GC. TLC is a thin layer chromato, LC is a liquid chromato, and GC is gas chromatography. Okay, there's the difference between all these 3 adalah dia punya stationary phase lah. Okay, and this technique of chromatography is that it is a versatile method. Okay, very easy to separate the components in your mixture. Okay, and we will have this we will learn this technique in our practical, the last practical. Okay, to separate the components of your mixture later. And then this technique of chromatography will help in identify the unknown, okay, by compare with the reference samples. Okay, and it's also no restrictions on sample type. It could be the organic sample, inorganic sample, biological sample, or medical sample, and it also high sensitivity. Okay, and for chromatography technique, okay, you must have these 3 essential components. You must know your phases. You have 2 phases. One is the mobile phases, second is the stationary phases. Okay, other than that, you must have your extract lah, which is the sample mixture, and then you will analyze your samples. Okay, so this example of GC chromatogram, the result by analyze using gas chromatography, you akan nampak different peaks, and different peaks ni kita nak compare, kita mesti refer to the standard. Okay, so we can identify the unknown peaks by comparison with the reference samples. Okay, chromatography is a physical separation method, okay, di mana kita perlukan that sample mixture, and the sample mixture or known as extract, okay, di mana kita akan run them on these 2 components, which is the stationary phase and another one is the mobile phase. Okay, what is stationary phase is which is a part where it is stationary, dia akan static, and however mobile phase is the solvent that will move lah. Okay, so usually solvent will be the mobile phase, okay, that will move, bring, ataupun dia akan carry through your mixture or extract. Okay, the stationary phase is the substance di mana dia fix in place, okay, for the chromatography procedure, and this phase, okay, to which solvent and the analyte will travel through or bind to. Biasanya benda yang where the mobile phases akan go through it lah, that will be the stationary phase. For example, kita nampak tadi paper, thin layer chromatography, the paper. So the paper will be the stationary phase, di mana the mobile phase will run through the stationary phase, bring the sample mixture to separate them to 5 different components of compounds. Okay, and the mobile phase is the analyte, okay, and solvent mixture which will travel through the stationary phase. Chromatography, okay, they have two phase, mobile and stationary phase. Okay, and this mobile phase could be a gas or it could be a liquid, okay, liquid solvent that passes through the column. Column biasanya berada pada GC and HPLC kan, di mana dia ada column that you have to buy, okay, inside the machine, and inside the column this is the part in which, okay, the mobile phase will bring your sample mixture to separate them. Okay, so the column, okay, and the stationary phase, it could be solid, liquid, or liquid supported on a, so biasanya column lah, column will be the stationary phase and the mobile phase will be the gas, or it could be the liquid that akan run through the column. Okay, stationary phase is the column, the mobile phase is the solvent, or that could be liquid, and also it could be gas. Okay. Okay, so this is a example of chromatography, separation by chromatography. The column here will be the stationary phase and the mobile phase will be the gases, or it could be liquid lah, bergantung pada types of machine. Okay, and this is a sample mixture. Sample mixture injected, di mana dalam sample mixture ni ada numbers of this is an extract yang kita yang di mana ada numbers of components or compound yang kita nak pisahkan. Okay, and then the mobile phase will sweeps the sample whether it sampai pergi ke detector. So ikutlah mobile phase ni apa, gas ke liquid tak pasti. Okay, kalau HPLC gas, liquid lah. Kalau GC gas lah, the mobile phase. Okay, and then the mobile phase will bring the sample sampai ke detector, and the detector dia akan detect the presence of compounds. So the separation process, okay, the sample components are carried by a mobile phase. Mobile phase akan bawa the sample mixture sampai ke detector, okay, along the stationary phase. Okay, so the individual species are retarded by the stationary phase based on various interactions such as surface adsorption, relative solubility, and the charge. Okay, so dia akan the components or the compounds yang akan ada berhenti pada stationary phase, okay, akan based on the surface adsorption, relative solubility, and the charge of the compounds. Okay, so it's in here we could see, okay, the the sample mixture injected, okay, through the column, through the stationary phase, and the mobile phase will bring the sample mixture, okay, and pisahkan components based on its adsorption, surface adsorption, based in its punya charge, okay, and then dia akan pisahkan mereka. Okay, so then kita akan detect them on detector. Okay, so stationary phase will stay put, the column, mobile phase dia akan float around, could be the gas ke ataupun liquid, according to machine yang kita guna. Okay, and how does it affect sample movement? Kalau stationary phase dia akan retain by surface interaction, and however mobile phase dia akan move the sample along. Mobile phase dia akan fungsi dia adalah untuk bawa along the stationary phase. However, for stationary phase dia akan retain the sample by surface interaction. Dia akan interact with the sample, okay, that brought by the mobile phase. Analog to an obstacle course, and this is a motivating force yang akan tolak mobile phase, and stationary phase yang halangan yang akan dia lalui oleh mobile phase. Okay, example of stationary phase is the silica gel, and mobile phase it could be the ethyl acetate. Okay. This is a analogy for chromatographic separation. This will be the kita punya stationary phase column tadi, okay, and this is the sample mixture terdiri daripada hornets and bees, mixed swarm of bees and hornets, okay, dia akan swarm enter the flower bed, okay, so dia akan visit the stationary phase, the obstacle lah yang ada pada stationary phase, okay, and then because of the obstacle dia akan pisahkan the mixture. So the hornets will leave the bed first and then follow with the bees. Okay, so that is the function of the stationary phase study, to give the obstacles to the sample mixture. Okay. Okay, now the chromatography terms, okay, that you have to know. Okay, first is the analyte. Analyte is the compound yang you want to separate lah from the mixture, from the sample mixture. Okay, it will be it's a substance which is to be purified or isolated during chromatography. Next is the chromatograph. Chromatograph is the machine yang di mana you run your sample mixture. Okay, yang ada mobile phases, yang ada stationary phases. Okay, that is machine. Okay, the machine, it could be the HPLC, it could be the GC. Okay, that is known as chromatograph. Then next is the chromatogram. Chromatogram is the visual output. Okay, the hasil of your chromatography technique. Okay, you akan dapat a graph. So that is known as chromatograph.